Hi, seventh graders. Hope you're having a good day. I am here to teach you about the lesson that you're missing because of MCAs and because of the play in the afternoon. So thanks for coming on to Moodle and checking out what we would have done had we been in class. So I'm going to walk you through the lesson. It might be a little bit choppy because I'm just going to kind of stop and go here, and there will be times throughout where I'm going to actually ask you to pause the video so that you can do something, maybe check out a link on my Moodle page or write something down on your paper. Okay, so last class, when I was also not around, you went with the sub on this page and in your packet. And one of the big messages, I guess, other than it was probably fun to look and see what traits you had and what traits your classmates had and how unique you were. But the big message is that if something is dominant, if a trait is dominant, it is not always more common. And that's a big misconception. If I pull up this here, this is some data from another year, a B-Day class. And if we look, for example, let me look here quickly. Okay, so for example, mid-digit hair, if you look right here at this bar, the mid-digit hair, having hair on your fingers, was actually the recessive form was more common. Now, but those two bars are pretty close. It's almost 50-50. But look at this one here, widow's peak. Not having a widow's peak is the recessive form, and apparently in this class, everybody had a straight hairline. No one had a widow's peak, showing us that just because something is dominant, for example here, widow's peak is dominant, but we didn't have anybody. Dominant doesn't mean more common. Dominant doesn't also mean better. Traits are just traits. There's different forms, and I've had kids over the years think, oh, dominant means better. Not necessarily. For example, let's also look at this. So on your packet, I think it's page 10, you can see some everyday traits that you either show the trait or you don't. So there's only two versions. So there's dominant and recessive, and you can see long earlobes and short, and maybe some other things you don't know, but I want to draw your attention to this right here. Having 6 or more fingers, 6 or more toes is actually dominant, but I don't see a lot of people with 6 fingers or 6 toes. So again, more dominant, or something is dominant, it doesn't always mean more common. All it means is that if you do get that particular gene, the dominant gene, you are guaranteed to show it in your phenotype. And that's all dominant means. Okay, so now for homework for today, which I won't be able to check off, I'll have to look at it when I actually see you sometime. So if you didn't get it done, you have a little more time to do that Rosalind Franklin reading, which is pages 13 and 14, I think. So you were asked to read about her, the female scientist who took a very famous picture. And let's actually look at that picture right now. So this is it. This was done using X-ray crystallography. It's called Photo 51. And what I'd like you to do right now is I'm going to stop, or I'm going to ask you to stop the screencast, and you will find a link to today's lesson, a video. It's a couple minutes long, showing you how X-ray crystallography works and how Rosalind took this type of picture and how this unique X shape actually indicated a helix, a winding staircase structure. So check out the video and see what you think, because it can be kind of confusing how a helix would result in this type of X shape for the picture. Okay, so now that you've watched the video about the X-shaped structure, the crystallography, and you've seen a little bit more about how Rosalind produced that picture, just want to recap with you about Rosalind Franklin. And just keep in mind, you know, that it was during World War II when she was trying to figure out what DNA looked like, and she took that picture, and without her permission, a gentleman by the name of Maurice Wilkins showed the picture to Francis Crick and James Watson. And those two gentlemen, Crick and Watson, determined the structure of DNA. Now, of course, the debate remains, would those two scientists have figured it out without Rosalind's picture? Would Rosalind have figured out what the structure was, and the adenine and guanine and cytosine and thymine and the actual molecules that were a part of it? The debate remains. We don't know. but you know, I think it's important to know the story about Rosalind Franklin, especially as a female in science during that time. She certainly did not receive much recognition, if any, for her contribution for a long time to our knowledge of DNA. Okay, so now let's get to today's lesson. So what I'd like you to do now is, again, take out that packet. Hopefully you brought it home with you. And if you forgot to bring it home with you, I posted all the pages for today's lesson on Moodle as well. So now in our packet, I'm going to open up mine here. I'd like you to go to packet page 15. And on page 15 at the top, it's called Mendel's P experiment. So I'm going to pull that up as well. Here it is. This is what it looks like. Okay. So for this activity, had you been in class, I would have had you in groups and you would have gotten a baggie, which I actually have to grab just a second. Okay, you would have gotten a bag like this with a bunch of Easter eggs and some stuff inside. Okay, now I'm just going to act it out for you and show you what you would have gotten. I'm going to give you the data so you can pretend like it's you doing the lab, but you're going to be answering all the questions, and I'll tell you when to stop the video so that you can have some time to think. Okay, so I'll just start reading at the top. It says, imagine you have two pea plants. One is true breeding and only makes black seeds. The other pea plant is also true breeding and only makes white seeds. If you were to cross or mate these two pea plants, what would their offspring, the baby pea plants, look like? Okay, so the idea of this lab is I don't expect you to know what's going to happen. There's probably a lot of words that you don't know. You probably don't necessarily know what true breeding means just yet. And the concept of plants mating is probably new. So for a lot of kids, they don't know how this happens. Well, it's pretty simple. Pollen is plant sperm. And so right now, with all of this pollen floating in the air, allergies are high. So a lot of us are, it's kind of weird to think, but myself included, allergic to plant sperm. And so either via wind, the wind can blow that pollen, and if it blows it through the air, it'll go from one plant and land on another plant, kind of the flower part of another plant. And by taking that plant sperm from one, it can land on the other. And there's actually an egg. Don't picture a chicken egg, but kind of this white, gooey egg structure deep inside the base of the flower. And so when that pollen hits the top of the flower, it'll slowly kind of go down into the base and then fertilize that egg. And voila, as time goes on, you get seeds, and those are baby plants. Now, wind can blow the pollen, butterflies, bees, moths, so there's lots of pollinators out there. So what we're just going to kind of keep that in mind as we work through this experiment called Mendel's P experiment, and it'll make more sense as to why it's called Mendel's in a little while. Okay, so like it says, I want you to predict, write out a hypothesis. What do you think the first generation, which we call F1, first generation of pea plants would look like if we had one plant that had only made black seeds and another plant that only made white seeds? So you're going to make a prediction. What do you think all the seeds for the baby plants are going to look like? So I'm going to ask that you stop the video now and make a prediction and write it out here. And don't cheat, because pretty soon I'm going to give you the data, and I don't care if your prediction's wrong, but I want you to go through the exercise. So make a prediction here. Okay, now that you are back, we are going to collect some data. So here are my Easter eggs, and I'm going to pretend that these are my baby plants that were produced as my black seeded parent and the white seeded parent mates. So as I look in my bag, I've got 8 eggs, and there are 4 that are called, there we go, F1. Okay, so I'm going to open up those 4 eggs. So I'm going to first find the 4 that say F1 on them for my first generation. Okay, so exciting. 1 egg, open it up. Ooh, there is, let's see if I can show you here, a black seed inside. So I have 1 black seed, and we're going to also think of these like pods. Okay, so I'm going to go over here and I'm going to put 1, or you could do an X. Okay, so I've got for pod 1, this is my pod 1, my first egg, 1 black seed. Okay, next one, another black seed, make an X. My next one, Okay, another black seed, and one more. Oh, all black. Okay, so I've got all black seeds, so I have 4 and I have 100%. Okay. Now, this is a bit surprising. I don't know how many of you would have guessed that they would all be black. Quite often, kids will guess 50-50. But very rarely do I have students say that they will all be one particular color. Okay, so then I want you to go down a little bit, and I'd like you to write an explanation right here. Why do you think this is? Why did we get all black seeds? What happened to the white seeds from the one parent? So write out an explanation here, pause the video, and take the time to do that. Okay, now we're on to step 2. So for step 2, we are now going to pretend that the babies from the F1, remember they all have black seeds, that's their phenotype. I'm going to start using those words again. Their phenotype is all black seeds. Now let's pretend those babies, those pea pod plants mate. Okay, so the pollen gets carried over and all of that happens. We get some new seeds. What color are they going to be? So I would like for you to make your prediction right here. What color do you think? So pause the video and write out a prediction. Okay. Now that you've made a prediction for the F2 generation, let's actually see what happens. So I'm going to grab my next 4 eggs and open them all up. So exciting. Oh, another black one. So I'm going to make a mark there. Oh, a white seed. White seed. Cool, that's new, that's good. Something interesting. Black. And more. Black seed. Okay, so we have 3 black seeds and we have 1 white seed. So we have 75% black and 25% white. So fill in your data table. Okay. Now I want you to think about why this is. What happened? How did we have the white seed trait reappear? What occurred? Even though both parents had black seeds, all of a sudden this white trait reappears. Why do you think we got that result? So I'm going to ask that you pause the video and write out an explanation. Okay, now we are on to page 16 of the packet, and you might be, kind of trying to process that data a little bit and make some sense of it. Well, you just acted out a very famous experiment that a gentleman by the name of Gregor Mendel did. And he did his with pea plants, real pea plants, and he looked at many, many traits for those peas. And he had data, the percentages were the same, where it seemed like a trait disappeared and then it reappeared again. And every time he did that, the F1 generation always showed one version of the trait, and then the F2, every time, 75% showed one version and 25% the other. And Gregor Mendel was a smart enough man to know that something was going on, that he needed to keep collecting more data and trying to figure out why these rules, these genetic rules kind of were occurring. Why was it always 75 and 25 for the F2 generation? So what I'm going to ask that you do now is you will find a link on the Moodle page to read these pages in your textbook, pages 214 to 218. You'll open that link. Read about Gregor Mendel, please. And once you have read about Mr. Mendel, I'd like you to answer all of your questions on page 16 of the packet. Okay, so hopefully you've given the questions on page 16 your best attempt. Next class, when you are there on Thursday, you'll have one of our 8th grade science teachers going over the answers to these questions with you, giving you a lot more practice with questions like different crossing of different traits, discussing Mendel's experiment in a lot more detail, because usually kids have quite a few questions, which is great. So even if some of these were kind of difficult and you aren't quite sure, again, just come to class, please, with them attempted. The teacher will be checking this page off and giving you a check in my gradebook if it looks at least completed and that you gave it a fair shot. So The only thing that I'd also like you to do, and then you're done with the lesson for the day, is you will see a link again on my Moodle page for a Bill Nye genetics video. I think it's about 20-25 minutes, so go grab some popcorn, get a snack, and get all cozy and watch a little Bill Nye